All right, so hello everyone. Welcome to another Clinician Engineers webinar. For today, we have Dr. Amparo from the Bioelectronics Laboratory at the University of Cambridge. We're delighted to host you here for the day, and I will let you take the stage for now. So if you could go ahead.
Yeah, thank you so much. Thank you so much for the opportunity to talk and to discuss about the opportunities and the challenges as well for developing a new way of therapy, which is known as bioelectronics, in the context of metabolism and in particular in the context of type 1 diabetes. So let me start. Wait, what's happening? Yes. So let me start by, for the non-expert public, basically explaining what metabolism is and what we known as my metabolic function. So to put it very simple, basically the metabolism is this set of processes that occur at the cellular level that convert food into energy that allow us to carry our daily activities. So these processes essentially include the breakdown of food into energy, but also the elimination of waste products, and also, for example, the building and the repair of tissues, the regulation of hormones, and so forth. So it is, of course, influenced by many factors such as the age, the gender, the genetics, the lifestyle habits, like diet, exercise. And then particularly diabetes. Diabetes is a metabolic dysfunction because it affects the body's ability to properly regulate this metabolism of glucose, which is a type of sugar that comes from the food that we eat. So in healthy conditions, when we eat, the beta cells in the pancreas release this hormone that's called insulin that allows our body to store and to use the carbohydrate, the sugar containing the carbohydrates. In type 1 diabetes, however, there's a disruption of these processes because there's an autoimmune destruction of the beta cells in the pancreas, and therefore this insulin is not secreted properly, which cause large and very risky blood glucose fluctuations. So this disruption in glucose metabolism, of course, has very serious implications, including, for example, the damage of organs and tissues, increased risk of infection, and other complications such as heart diseases, retinal diseases. So this is a very serious disease. So metabolic diseases like type 1 diabetes have traditionally been treated pharmacologically. So what does that mean? For example, for type 1 diabetes, the traditional treatment includes measuring the glucose levels using a finger prick, or recently the flash or the continuous glucose monitoring sensors, and then injecting the insulin manually accordingly through these boluses, as we can see in the slide. But in the last years, we've seen the artificial pancreas, which is this closed loop system that responds automatically to changes in glucose levels by adjusting the insulin dose that is delivered. So the first commercialized system, which is the Medtronic 60G, it's the first hybrid system. And we can see in the slide on the top one how the normal glucose fluctuation over, like, on a day for a healthy volunteer look like. And in a person with type 1 diabetes that has implanted this insulin pump in the bottom graph, we can see that it really increases the amount of time of glucose within the target in green. But this is still far from the optimal glucose regulation that we observe in healthy individuals. Why? Well, these discrepancies probably arise from the very complex network of mechanisms that are involved in controlling glucose metabolism in healthy individuals, where we find a large number of hormones, metabolites, but also neurotransmitters that interact with each other to ensure a robust and a tight regulation of blood glucose fluctuations. But this complexity is not unique for glucose regulation. So regulating metabolism is a vital function that requires the coordinated activity of many physiological systems. So as a result, we see this complex neuroendocrine and neurometabolic circuitry that is involved. And we have regulation through hormonal circuits, such as, for example, through ghrelin, through insulin, through leptin, that are these primary hormones that mediate the sensation of hunger and satiety. But then we also have the regulation through direct neural circuits. So we have both the sympathetic and the parasympathetic circuits that influence the metabolic functions. So, for example, energy expenditure or the circulating levels of glucose in blood. And for example, research and scientists have shown that a dysfunction of the sympathetic nervous system may contribute to the pathophysiology of obesity and diabetes, as they've seen that a high hyperactivation of those pathways, neural pathways. So in particularly, the central nervous system, and therefore the brain, is an integral part of the regulation of metabolism. How? Well, by directly sensing the metabolic states and also releasing these neuroendocrine signals. So we know that the central nervous system communicates with the rest of the body, with the visceral organs, through both cranial and spinal nerves, both afferently and efferently. So this bidirectional communication. And at the level of the brain, we have the hypothalamus, which is one of the most important regions that are involved in the neuroendocrine function. And within the hypothalamus, we find these certain regions and populations of neurons, such as the arcuate nucleus, the ventromedial nucleus, and the lateral hypothalamic nuclei, that have a really specific role on these metabolic processes. And each of these nuclei, and within them, each of the different population of neurons, they are involved in different aspects of glucose regulation, from insulin release or hormonal release to the action of that insulin in the tissues, which is known as insulin sensitivity, and so on. So the question here is, can we interface with the nervous system through neuromodulation to potentially restore this dysfunctional neurometabolic circuitry, and also to prevent the progression of metabolic diseases? So right now, there are commercialized devices that are both invasive and non-invasive, that are being used currently to use different, like many other chronic diseases, such as Crohn's disease or rheumatoid arthritis, that I feel they can potentially be leveraged for patients that have dysregulation in metabolic control. So, for example, deep brain stimulation can be used to control appetite, energy expenditure, and the glycemic index through neuromodulation of the neurometabolic circuitry by targeting these specific POMC neurons in the arcuate nucleus. There are other studies of Parkinson's patients that had DBS implants that also were found to have improved metabolic control. And there's also ongoing research in stimulation of the vagus nerve to restore dysfunctional metabolic activities. So, for example, the vagus nerve stimulation can help to regulate blood sugar levels and appetite, leading to improved glycemic control and also weight loss. And in particularly related to this, there are many studies that have shown that vagus nerve stimulation can be very effective in promoting weight loss in people with obesity. So we see that bioelectronic medicine shows a huge potential for patients that have these metabolic conditions that may not respond properly or as good as we would like to these traditional pharmacological therapies. And of course, there's more research needed to understand the potential of vagus nerve stimulation for metabolic disorders, but these early findings suggest that this can be really a promising option. So in this context, a few years ago, I started to work on a novel system that goes beyond the purely metabolic systems by interfacing with the nervous system through both recording and stimulation, resulting in this type of neurometabolic closed-loop system that we called Neurometabolic. So the hypothesis here is that by replicating in more detail the physiological metabolic scenario that we can see in healthy individuals, then we can get better control of glucose levels. And this approach is very, very impactful because it can be used to treat other type of metabolic dysfunctions in addition to type 1 diabetes, and also like be leveraged in other chronic diseases as well. But what challenges do we face to create such as neurometabolic closed-loop system? Well, I feel that this is a kind of a puzzle of challenges. And in my lab here in Cambridge, at the Bioelectronic lab led by Professor George Malyaras, we are trying to contribute to solving each of these challenges. So I'll go one by one and explain also what we do in our lab. So of course, the first one is the monitoring of biomarkers. So in general, closed-loop systems require reliable sensors for monitoring metabolic parameters in real time. And of course, technical limitations in this sensor technology can limit the effectiveness of this closed-loop system. So currently, there are very limited biomarkers that can be used to accurately measure the metabolic processes in real time. So the only biomarker that right now can be measured continuously is what we say, like glucose. And it only provides a very limited information about the complex metabolic processes that are happening in our bodies. So we can combine this with other metabolisms like metabolites, as ketones. And there are now being developed some continuous monitoring systems for ketones, but these systems are now like a bit prone to, or maybe prone to inaccuracies, especially during like very sharp changes in blood glucose levels, in blood levels. So and they're also very expensive, so that limits the use. And in addition, another challenge that is associated with current continuous glucose monitoring systems is the potential for delays in the readings provided by the sensor. Why? Well, this is because basically the sensor is implanted in the to measure in the interstitial space, and therefore it takes some time for the glucose to actually go from the bloodstream to the interstitial tissue, and this of course can result in a lack of real-time or very accurate real-time accuracy, and that make it may make it difficult to make very immediate decisions based on the glucose readings. So to address these challenges, the researchers are exploring like new sensor type technologies, new algorithms that can improve the accuracy of the diseases. But overall, the lack of these reliable biomarkers can make it difficult to monitor and to adjust neuromodulation interventions in a closed-loop system. But still, researchers are looking into that. Now, the second part, which is more related to what we do in our lab, it's basically trying to address the challenge of the mechanical mismatch. So it's the mechanical mismatch between the neural tissue and our implanted technology, our implanted devices. Why? Because this mechanical mismatch can cause a lot of problems such as inflammation, scarring, and ultimately, of course, the failure of the device. So the reason behind this is that most of the biosensors or the interfaces that we implant are based on very stiff electromaterials such as silicon, such as carbon fibers that are, as we can see here, in the order of 100 of gigapascals. And the Young's modulus is around that order, whereas the brain or the nervous system, it's in the order kilopascals. And that basically causes this mechanical mismatch. So a promising strategy for that is to use what we are developing in our lab, which are these conformable polymeric-based bioelectronic devices that are made of substrates such as PDMS or parallel C. And they reduce this mechanical mismatch between the neural tissue, and their electrical properties can be readily tailored and to match those of the tissue. So in addition to that, what we can do is to add an extra layer of conductive polymers such as PSS on top of the recording sites to further reduce the impedance, so that electrical resistance, and also reduce the mechanical mismatch with the tissue. So these are the type of devices that we are developing in our lab and that we are implanting preclinically. So all the work that we do is preclinical. That's something important to highlight here. So now the next step, once we have basically been able to record something from the nervous system, from the peripheral nerves or from the brain, we need to address the fact that our neural signals may come from multiple sources, so multiple neurons, multiple populations, and that requires several factors. So for example, one of the things is the spatial resolution when recording. So we need to understand what's the spatial distribution that we are looking for in our neural signals to provide valuable insights about their origin, but also their function. So for example, in the brain, we have high resolution imaging techniques that are being developed, such as functional MRA, that can be used to identify the specific regions that are being activated. But in the nerves themselves, we can use something that are called multi-electrode arrays or MEAs. That these are high density multi-electrode array caps, or also they can be penetrating into the nerve, which are called as intraneural electrodes, such as the time or the life electrodes. And these can increase the spatial resolution for both recordings and also stimulation. But secondly, it's very important to identify the source of the neural signals when we are decoding from them. And this requires normally advanced signal processing techniques that can distinguish from signals originating from different sources. So for example, we have the group of Dr. Dominic Durand that had made extensive work on using blind source separation algorithms to be able to reconstruct the sources. And in my case, I've been exploring the use of data mining techniques such as correlation analysis towards this objective. Then, of course, we need something that integrates both the metabolic and the neural information and enable to enable that control action that we want to do. In this case, either it's another stimulation or injecting the insulin accordingly. So of course, these type of control algorithms, we need something that is individualized, because each patient is going to be different, but also we need a very fast online processing. But in addition to that, in the particular case for glucose regulation, we have metabolic processes that occur in a very slow temporal scale. So like changes take place around minutes or hours. But in contrast, we have these neural signals and neuromodulation processes that occur on much faster timescales. So changes taking place in the order of milliseconds to seconds. So these different timescales for the metabolic and the neural dynamics basically highlight the need for a very highly adaptive and a flexible closed-loop system, a flexible control algorithm that can integrate and respond fast to these signals across multiple timescales. And then finally, so we've seen that metabolism is a very, very complex system where basically we have multiple interrelated pathways and feedback loops that makes it very challenging to accurately model and to control these processes. And we've seen that we are working towards addressing this, but we have to bear in mind that the relationship between the neural activity and the metabolic processes is very complex and is still not fully understood, despite we're progressing on that. And that's why, like right now, it is very difficult to develop a precise and an effective neuromodulation strategy for metabolic control over the long term. However, we are doing all these foundational steps towards basically putting all the pieces together and develop this type of closed loop technologies for improving metabolic control. So I hope that with this talk, I have been able to transmit the encouraging horizon that bioelectronic medicine promises to provide improving control and of course, like through overcoming these current limitations of traditional approaches. And with that, I would like to finish by thanking my colleagues at the Bioelectronics Laboratory, of course, my supervisor, Professor Josh Maliaras, and in particular, Dr. Alejandro Castiner Lombarte, who has been real support on this. And of course, I also want to thank the funding bodies that are allowing me to develop this research in Cambridge. So special thanks to the 1851 Research Fellowship by the Royal Commission of the Exhibition, Royal Commission for the Exhibition of 1551, and also the L'Oreal Foundation in UK and Ireland. Thank you so much, and don't hesitate to reach me at this email address if you have any other questions or any comments that you would like to discuss with me. Thank you so much.
Thank you, Dr. Amparo. So we have a few questions in the chat.
Brilliant.
Yeah. And if you'd like to address a few, we have a question from Dr. Banu. So she's asking, what are some of the challenges and limitations in the field of bioelectronics, and how are researchers and engineers working to overcome these challenges?
Okay, well, I hope that during my talk I've been able to give a kind of a broad overview of the most important challenges that we are addressing right now. In particularly for bioelectronics themselves, it's basically building these new devices that cause a minimal interaction or a minimal impact to the biological tissue. So it's finding the materials that meet the mechanical and also the electrical properties to interface, in this case, with the nervous system. But also we need to develop all the electronics and everything to make it a full implantable device. So that's something that is also not easy. We don't want things to be connected, so we want things to be wireless, and all those added features add more complexity to the problem. But that's something that specifically, like what materials, what configurations, like how, like what design do we need to build to be able to interface better with our bodies. It's something that we are working very hard in this lab, in my lab, the bioelectronics lab in Cambridge.
That's wonderful.
So, thank you so much.
All right. Well, I really enjoyed your talk. I have a few questions myself, but there is another question we have in the chat. So
Perfect.
Dr. Vaughn is asking, she's interested in knowing whether a bioelectronics study on glucose monitoring has translated into an actual product in this industry. And she says, thank you for this amazing presentation, Dr. Amparo.
Thank you so much. So if we are talking about bioelectronics for glucose sensing, yes, because essentially like all those glucose sensors are based on bioelectronic technology. If we are talking about bioelectronic devices to interface with the nervous system to decode metabolic information, this is something that, of course, it hasn't been translated into the industry now because it's being under, as I was saying, very preliminary or foundational research. And this is something that my research is focusing on right now. Like how can we leverage these bioelectronic systems to interface with the nervous system to be able to capture metabolic information or information from the glucose levels. And this is also something that the Feinstein Institutes in New York, they have been pioneers on. They published a paper on using the vagus nerve as a glucometer. So that's very promising, but of course it's really far from actual translation into the industry or into the clinic as well.
All
I hope
right.
that answers the question.
Yeah, I think it does based on the chat.
Perfect. Thank
Great.
you.
So I have a question myself, if you don't mind.
Of course.
So in terms of bioelectronics, have you found that there is a lot to do in terms of hardware, or is it mainly a software domain? Are there
Yeah.
certain languages that you thought were useful in this field, useful to learn for somebody trying to break in, or is it more of a mechanical or electronic engineering kind of thing?
So that's a really good question, and that's something that I probably have not highlighted enough in the presentation. But this type of work is extremely interdisciplinary, extremely. So it's not only the progress in one single area that will make it work. It's probably the progress and the contribution of like small steps in parallel in all these fields that will allow to build this complete system. So of course, we need to focus on the hardware side, so on these bioelectronic devices, on the electronics to communicate the transmission and the batteries as well. So batteries for stimulation is a very, very important limitation or issue that we need to address. But of course, as you were saying, like once we have that data, what do we do with that? So of course, we need to develop in parallel all these algorithms that allow us to extract meaningful information and develop like the control algorithms as well to be able to define what to do with that information that has been collected, right? So it's I wouldn't, so in my particular work, I address a bit of everything, and I don't feel that leaving one bit behind will help at all. So everything has to be progressing parallel. And in terms of what you were asking about, like what language or coding languages would be good for someone that would like to get involved into this, I would suggest that, so every researcher has their own coding language that they feel more confident on programming. It's true that we may be lacking a standardized way of developing these algorithms, and probably there's a lot of replication and duplication of the effort on developing these algorithms. But right now, for example, in my case, I do use Python, but I know there are researchers that use R, they use MATLAB. So hopefully in the future we can find a standardized a strategy to standardize all these different strategies to address the coding problem. But so far, I don't find that there's one that is particularly better than the other. I feel it's a matter of the researcher to select the one that they are that they feel more comfortable with.
Okay, well, I mean, I'm kind of glad to hear that because then that makes it more accessible for anybody interested in the field to try and see how they can apply what they're familiar with, or the background that they come from.
Exactly, and something that I feel it's very important is people from different backgrounds, if they see this type of interdisciplinary research, they may be willing to apply their own expertise, even if it's in a completely different field or application.
Mm.
They may be able to apply their knowledge into these types of, in particular, health applications, because in the end, science is science, and the techniques and the strategies are there to be used in different problems. It's what we are probably lacking in the scientific communities that, well, we are not lacking it, but hopefully having a more interdisciplinary, but not within the same discipline, but really interdisciplinary research will allow to progress more. And biomedical engineering and electrical engineering are two specific fields where that is specifically relevant and where that is particularly it's particularly visible. So we do see that a lot.
Yeah, medicine and engineering. All the way.
Yeah, exactly.
Yeah, well, that certainly really, it really kind of laps in with the clinician engineer's mission aim in general. So yeah, we're really glad that we could have you here today for this talk.
Oh, thank you so much. And of course, like I don't want to leave without mentioning that this type of research doesn't make sense if it's not developed with the help and the continuous guidance of clinicians, of patients, of basically the people that are gonna use this and that are gonna need it. So if people that are seeing the webinar, if you feel that you are related to the field, and please contact me, because I'm more than welcome to discuss my project, my research, and I'm open to suggestions on how to guide it to basically have the impact that I want, like really improve the quality of people's life. So